Hello, my name is Ayman Al-Waqsa, and this is my research project for the summer of 2024 in Dr. Maria Basil's lab in the Lung Biology Institute. Um, the main focus of my research project is a novel cell type called RAS cells that were discovered in 2022. RAS, or respiratory airway secretory cells, are progenitor cells that are newly discovered and were found to have um, progenitor-like ability for AT2 cells and are predicted to have that same progenitor-like ability for goblet, ciliated, and neuroendocrine and AT1 cells. Uh, Not only were these cells found to play a vital role uh, in the creation of new cells, but they're also found to play a vital role in uh, diseases like COPD, or chronic obstructive pulmonary disease, which is the third leading cause of death worldwide. In COPD, uh, or these cells were found to be, found to be in the injury site for COPD, which is the respiratory bronchioles. Uh, these cells are only found actually in the respiratory bronchioles of mammalians, and as shown in the poster in front of you, uh, mice do not have those respiratory bronchioles, um, and are not predicted to have RAS cells. Um, these cells, my project really tries to explore the importance of notch signaling for maintaining RAS cell fate and function. And to do this, to test Notch's importance, we had to introduce a new interface into the lab, a new technique for cultivating these cells. This relatively new technique is called the ALI, or the air liquid interface. And essentially the premise is that these cells, instead of being in a 2D culture, they would instead be in a 3D culture where one side of the cell faces media and the other side of the cell eventually gets airlifted and faces air. So it faces oxygen, CO2. And the theory behind this essentially is that this would better mimic uh, the, the the results from experiments that use ALI would better mimic the human, or would be more translatable to the human lungs.